So next, I'm delighted to introduce Markel Gómez Letona, who's going to talk to us about respiration in the interior ocean. So Markel, over to you.
Okay, sharing my slides. Can you see them?
Yes, I can.
Okay, so well, yeah, my name is Markel. I work as a postdoc at the Laboratorio de Geoquímica Orgánica in the Instituto de Investigaciones Marinas. And today I'll be showing you the work that I have done alongside José Antonio Álvarez-Salgado, well, basically quantifying respiration rates in the mesopelagic ocean. So this work is part of the Ocean ECU project, which aims to improve our understanding of the role of the oceans in the global carbon cycle, and has an important focus on all the processes and pathways involved in the biological carbon pump, which you probably already know, but it's a concept that groups all the processes and pathways, as I said, that contribute to transport organic carbon from the surface ocean, where it is fixed by phytoplankton, well, from there to the interior of the ocean. And among the processes influencing this biological carbon pump, we have an important one, which is respiration by heterotrophic organisms, which consume part of this organic matter that is being transported downwards, and thus attenuate the carbon flux to the interior of the ocean. So here I show sort of a simplified diagram of the main three pathways involved in the biological carbon pump, which are the well, sinking particles, also the migration of, or the vertical migration of organisms, some zooplankton and fish, and also the physical transport, the subduction of dissolved organic matter and also suspended particles to the interior of the ocean. And to give you an idea of how much respiration can influence these vertical fluxes of carbon, from the roughly 50 petagrams of carbon per year of net primary production in the ocean, only around 4 to 12 petagrams of carbon get exported each year out of the epipelagic layer. So already a good chunk of it is respired within the epipelagic layer. But then within the mesopelagic, which goes from 200 to 1,000 meters roughly, also a significant fraction of it gets respired. And then, of course, then in this way it influences how much it can reach the deep ocean. So in that regard, this mesopelagic layer acts not only controlling, or I mean, I should say the respiration happening within this mesopelagic layer acts controlling not only how much carbon will return to the atmosphere in the short term, but also how much it reaches below 1,000 meters and thus becomes available to be used by deep ocean communities. So in this regard, as I said, it's sort of a key gateway between this sort of more surface layer where this carbon is fixed and the deep ocean where the residence times of this carbon are low. So within the mesopelagic, the organisms that are responsible for the bulk of the respiration are not visible to us. So most of the times, the mesopelagic will look like something like this. But if we look closer, we will see that it is inhabited by millions of microbes, and chiefly by prokaryotes, like this shown in this microscope image. So, as I said, these prokaryotes are the ones responsible for the bulk of the respiration in the mesopelagic. And as they respire, of course, they consume oxygen and organic matter and release carbon dioxide. So they are responsible for the gradients and changes in oxygen and also carbon that we observe below the mixed layer. And hence, if we were able to estimate the changes of oxygen over time in the mesopelagic, we would be able to have also an estimation of the respiration rates that are being done by these heterotrophic bacteria. So, of course, studying respiration is nothing new in oceanography. It has been done over the last couple of, well, more than the last couple of decades, as summarized in this timeline with some relevant works. Most of them have been done studying respiration in specific regions of the ocean, So they did not provide a global scale in terms of at which scale these rates were represented. In recent years, we have examples like the ones by Sulpice and collaborators, where they did have respiration estimates with a global scope. But also in these cases, the areas represented by the profiles of respiration they estimated were quite big. So for example, they had one profile for the entire subtropical North Atlantic. So when setting out to do this work, the main goal we had was to be able to provide some estimates of respiration which had a global scale, but at the same time that they had some sort of spatial resolution, which would potentially allow us to see some variability within this respiration in the mesopelagic layer, and also potentially be able to associate this variability to some biogeochemical and oceanographical processes happening within this layer. So as I said before, we wanted to do this by measuring how oxygen changes within this mesopelagic layer. And of course, for that, we need, on the one hand, oxygen measurements, but on the other, some sort of variable that gives an idea of how time passes. So for that, we have followed previous works and used transient tracer gases, and specifically CFC11, CFC12, and SF6 to compute the ages of seawater samples. We use these tracer gases because they have well-known source functions. They are conservative, meaning that they do not interact with biology. On top of this, the natural sources in the ocean are not significant, and they have residence times that make them appropriate for the timescales that we need to estimate respiration in the mesopelagic, meaning that the residence times are adequate for processes going from multiple years to decades. So very briefly, the way we estimate the ages is taking values of concentrations of these tracer gases in seawater, computing their partial pressures, and then matching these partial pressures with the values of these gases in the atmosphere, which are well characterized, as shown in this figure. So if we take the difference between the date we obtain by matching partial pressures, that date is the last time a water sample was in contact with the atmosphere. And if we subtract this difference to the date in which the sample was collected, we obtain the age of the seawater samples that we have. So to have a global scope, we use the GLODAP dataset, which I'm sure most of you already know about it, but we basically filtered, of course, samples which had oxygen measurements and at least one of these tracer gases. We estimated the ages and then kept only those which for dates which had like at least some reasonable uncertainty. And then after all this filtering, the samples that we kept are shown in this map. For the CFC samples, this meant keeping roughly around 90,000 of them, and for the SF6, close to 20,000. Then we grouped these samples according to the water mass in which they were found. We defined water masses based on definitions from the literature, taking into consideration the geographical extent and also the density ranges. And we grouped them into three groups by roughly their depth. So we have central water masses from 150 to 500 meters, intermediate ones from 500 to 1,000 meters, and deep ones from 1,000 meters to 1,500 meters. In total, 34 water masses. So in terms of the actual estimate of the respiration, in terms of oxygen utilization rates, instead of using the absolute values of oxygen, well, once it is computed as apparent oxygen utilization and the ages, we first did like a simple correction to remove the variability associated to the mixing of these water masses, because we were interested in the variability associated to biogeochemistry, basically. So what we did was to do multiple linear regressions with potential temperature and salinity and these variables as shown in this slide, and kept the residuals from these regressions, which are here indicated by this delta symbol. And then it was these residual values, these values from which the variability associated to water mass mixing has been removed, that we use to estimate oxygen utilization rates with these regressions of apparent oxygen utilization versus age. The slope of these regressions is what we are interested in, as it constitutes the oxygen utilization rate. So, as I said before, we were interested in having some spatial resolution for these estimates. So we divided the water masses into a degree of, into a grid of pixels of 10 degrees, as shown here. So we split all the water masses in these pixels and then estimated these regressions within each pixel, as shown in these examples. So then we kept the regressions that were positive, that which were basically all of them, but then filtered also keeping only those with an R-square above 0.15 and p-value below 0.001. So in these examples, the top row would fulfill this criteria and the bottom row would not fulfill it. So we kept only those that fulfill this criteria, as in the top row. So basically, the result of applying all this approach to the global dataset is shown here, which is like the main figure that summarizes the results. Again, I have grouped the results based on the location of the water mass. So the central water masses are the ones on the top row, then the intermediate water masses, and the deep water masses on the last row. And we repeated the analysis for each of the 3 tracer gases that we use in the analysis to just not rely on a single one and be able to compare the results that we obtain depending on which tracer gas we use. So in general, the oxygen utilization rates ranged between 0.6 and 27.3 micromoles per kilogram per year, which is a range that falls within the values that have been reported previously for the mesopelagic layer. So, but then also we can see that, as expected, there was a decrease in respiration rates with depth, going from central, intermediate, and deep water masses. In these box plots, I summarize the results per water mass specifically, and as you might already be thinking, not only from this plot but from the previous maps, there was variability not only across water masses but also within water masses. And in this regard, we wanted to highlight 2 of the main results that we could summarize from all this data. The first one is regarding the central water masses, which I show here plotted against latitude. As we saw that these rates tended to be higher with decreasing latitudes, meaning that respiration in these central water masses was higher closer to the equator. And we compared this to the proxy we estimate from satellite measurements, which I show here in this plot on the right. And we observed similar patterns between these carbon flags and respiration estimates, which makes sense because these central water masses, as you might recall, are the ones closer to the epipelagic layer, and hence they receive not only a higher quantity of carbon flag by the mere fact that they are closer to the epipelagic, but also they are more closely tied because they are, again, physically closer to the place where this carbon is produced in the first place. This showed actually correlations that were significant for all oceans, Atlantic, Indian, and Pacific, but were particularly stronger for the India, with R squares that were over 0.4, which for a single variable was quite significant. And then we also wanted to highlight that actually, contrary to what we expected, within this specific layer, there was not a significant relationship with potential temperature, which was surprising because as a metabolic rate, respiration is also dependent on temperature, but here it seems that there's like a higher or a stronger control by the carbon input that this layer receives. So then going into the intermediate and deep water masses, in this case, the patterns that we observed in respiration were more associated to the circulation and aging history of water masses, of these water masses, than to the vertical flux of carbon that they receive. So I want to illustrate this with two examples. The first one is the one I'm showing here, the Antarctic intermediate water in the Pacific. This water mass is formed in the southeastern Pacific, as shown in the map on the bottom, in this like dashed box, and then circulates toward the northwest and then toward the south. And we could see that there's a trend of decreasing values also in these rates from its formation area towards the northwest and then to the south. Implying that as this water mass circulates and the organic matter pool within it gets transformed and utilized and gets or becomes more recalcitrant, the respiration rates of the heterotrophic bacteria within this water mass also decrease. Another example illustrating this is the case of the North Pacific intermediate water, which is formed in the Northwestern Pacific, specifically in the Sea of Okhotsk in winter. As this water mass is formed and it ventilates this depth range in the North Pacific, as can be appreciated on the map on the right by the high oxygen concentrations there. But also it carries with it newly formed dissolved organic carbon. And on top of this area is also one which receives significant amounts of carbon by the sinking flux of particles. So it's this formation area, or like the region close to it, is one in which the bacteria in this water mass receive or have available a high amount of labile carbon, and hence or what we think is that explains the high rates of respiration that we observe and that I highlight here. And then as this water mass spreads towards the rest of the North Pacific, again, this organic matter pool would get utilized and the organic matter pool as a whole would become more recalcitrant. And this would come along with, again, reduce respiration rates within this water mass. And then just to going with the end of my presentation, I just wanted to compare our results with previously reported values. So in this plot, I summarize our rates for the Atlantic, Indian, and Pacific Oceans, which are shown as sort of average smooth profiles with the color ones. So the golden, pink, and blue ones are our oxygen utilization rate profiles for each of the tracer edges. And then the compilation we did from across 55 studies with more than 600 values is shown as gray dots, beg your pardon, and their average profile is the black line. So we see that overall they were in quite a good agreement, particularly in the upper mesopelagic, despite the well, first of all, the different approaches that were followed in these studies, and also the variability in the regions that they studied. But as I said, there was good agreement, and it was particularly so with the rates based on SF6, which agreed quite well also in the lower mesopelagic too. And this database, in case someone would be interested, is deposited in Zenodo. So this compilation of rates from the literature can be accessed openly if you search for it. Also, if you follow the QR code that is shown in this slide. So just to wrap up my presentation, I just wanted to highlight the take-home messages. Going back to what I was describing as the motivation for this work, we have provided with this quantification of respiration rates in the mesopelagic, some estimates that are both global in scale, covering basically the entire global ocean, but also at the same time having some degree of spatial resolution that allows us to at least discern some patterns and also potentially associated to some both biogeochemical and oceanographical processes. Specifically, as I said, the central water masses presented oxygen utilization rates, which were related to the vertical carbon flux that we estimated, suggesting that there was a control by, and in turn, a control over the biological carbon pump by respiration. And on the other hand, in the case of the intermediate and deep water masses of the mesopelagic, the oxygen utilization rates in these water masses were more associated to the circulation and aging history of these water masses. As I said before, because we suggest that as these circulate and get older, the organic matter pool within them gets more recalcitrant, and hence the respiration rates of these heterotrophic bacteria also decrease or coupled to this aging process. So yeah, this is basically it. If anyone would be interested in getting into more detail about this work, it has been published. You can follow this link, or again follow the QR code. It is openly available, so anyone could read it if they are interested. So yeah, that's basically it. So if there are any questions or comments, I'll be glad to take them.
Excellent. Thank you very much. Very interesting. I've spoken to a few people relatively recently about estimating respiration from the biological side, so it's very interesting to see a perspective of using sort of traces like age and things like that as a way of estimating it. Yeah, very nice. So as before, do people have any questions? Please put your hands up or type into the chat if you've got a question for Markel. I'm sure we'll have something eventually. So just to get started then, I thought the result that you found of the link between the respiration rates and the carbon flux was interesting, particularly the fact that you didn't seem to find a relationship with the other factors people tend to think in conjunction with that, like temperature and oxygen. Do you have a feel for what else is at play there? Is it community which is important, or is it just a case of everything's the same everywhere, and so the rates don't tend to change?
In terms of that, we don't really have like community composition data or something like that we could compare. So it's just like in terms of the amount of carbon that can reach those depths that we could like at least try to estimate the relationship. And then in terms of the relationship with temperature, yeah, I mean, I wanted to say that within this specific layer, we did not find a relationship, but within taking into account the whole data set that we generated, we do find a relationship with temperature, but it's like a wider temperature range, which also involves intermediate and deep water masses. Because we are actually now trying to also produce at least some parameterizations of these rates in terms of, first of all, of thermodynamics, so getting activation energies of these rates. And we do find significant relationship with temperature, although it is true also that there's some quite variability within that, so there's quite some noise, but there's a relationship, but not within this specific layer. So our idea is that the main driver, at least what we can see, is the carbon flux they receive and they use as substrate to perform this respiration.
Okay, but that's an interesting qualification that you mentioned, the fact that you do sort of at a broader scale, you still find a relationship with temperature. Okay. Oh, yeah. Very interesting. Thank you. So hopefully it's given people time to think and phrase any questions. I can see there's something in the chat. I'm not sure Well, I think that,
Otherwise, they could send an email if they want. If they come up with questions afterwards, then
yeah,
they send an
the
email.
chat was somebody just apologizing they had to leave. So unless anybody has any last questions. So as Michael says, please do get in contact with him if you do have any questions. I'm sure he would be delighted to hear from you. Otherwise, thank both of our speakers, Martha and Michael, very much for two excellent talks. And hope to see many of you on the next call. Actually, that's in a Pacific time zone, so that may be challenging for some, but catch the recordings. Check out the YouTube channel. There are lots of talks there. And if you want another chance to go over Martha or Michael's talk, then please do take a look. Otherwise, thank you all very much for coming.
Thank you.
Thank you.
Bye.
Bye.
Thank you.